All right, good afternoon or morning, wherever you might be, and welcome to another webinar sponsored by NAVBO, North American Vascular Biology Organization. My name is Ondine Cleaver, and I'm current president of NAVBO, and I'm very pleased to be introducing today's session. Today, we're pleased to welcome our speaker, Dr. Courtney Griffin. She will present research entitled, "What Chromatin Remodelers Can Teach Us About Vascular Development and Integrity." So by way of background, Dr. Griffin is currently a member of the Cardiovascular Biology Research Program at the Oklahoma Medical Research Foundation, or OMRF, and an adjunct professor at the University of Oklahoma Health Science Center. She's also the scientific director of the Oklahoma Center for Adult Stem Cell Research. In her training, she did her undergraduate degree in biology at Harvard College in 1995 and her PhD in biomedical sciences at the University of California, San Francisco in 2001. During this graduate training, she became interested in vascular development and its implications for embryonic growth and for postnatal health and disease, and she pursued her postdoc training at UNC Chapel Hill, where she was exposed to the field of epigenetics. And in 2008, she launched her own lab at OMRF in Oklahoma City, studying the impact of epigenetics on vascular development. So we're really excited to have her speak today. So at this time, I'd just like to welcome Matthew Menendez. He's a postdoctoral fellow also at OMRF, and he is going to monitor today's questions. So the way we're going to handle this is in two ways. Throughout the presentation, you can type your questions into the question box, which is on the control panel, usually shows up to your right there on the screen. And these questions will be answered at the end of the presentation. Mr. Menendez will compile these and pose the questions to Dr. Griffin at the end of the presentation. At the end of this question and answer period, if we have time, attendees will be able to ask their own questions live in real time and talk with Dr. Griffin. So if you have a question, please raise your hand by clicking the hand icon on the left side of your control panel. You'll be recognized, your mic will be unmuted, and then you'll be able to ask your question live. So I'll point out also that at the bottom of this control panel, there's a list of references provided by Dr. Griffin that are available in the handout box. It's a PDF, and there's also a chat box at the very bottom. So if you experience any technical problems, reach out. You can also click on the Help tab at the top of the control panel and scroll to the bottom to the Help screen for the GoToSeminar technical support phone number. Hopefully none of that will happen though. So please note that this webinar is being recorded and archived on the NAVBO website for future use. And so without further ado, let's all please welcome Dr. Griffin. Thanks for being with us.
All right, thanks, Ondine. I appreciate the introduction, and thanks to everyone who's tuned in today for taking time out of your day to participate in this webinar. On a personal note, I just have to say I sincerely hope that you and your families, your friends, your lab mates are all staying really safe and healthy during this terrible pandemic, and I hope we can get back to scientific normality somewhat soon. So I'm really pleased to have the opportunity today to talk to you about a somewhat unconventional approach that my lab takes to study vascular development and integrity. I know that these are topic areas that many of you study in your own labs. And I should point out that the reason that we are particularly interested in studying these topics and the angle that we take to study these topics is that we focus on the genetic regulation of these processes. And we think that this is critical, as many of you do, because we believe that our ability to manipulate genes that control vascular development and integrity will help us to promote desirable processes like embryonic survival and wound healing, and of course, to inhibit undesirable processes like tumor growth, thrombosis, or lethal hemorrhage that can occur when you have a ruptured aneurysm. So with this in mind, I should point out that historically, as a field, we have often identified genes that are important for these processes using conventional and very classic genetic techniques, including reverse and forward genetics. And of course, reverse genetics refers to the mutation of a specific gene in order to look for a phenotype of interest, in this case, a phenotype impacting vascular development or integrity. And forward genetics involves the mutation of a whole organism to generate those same phenotypes, and we're of course interested in vascular phenotypes, and then to backtrack to identify the gene that led to those vascular phenotypes. And these are really terrific tools, and I should point out that when I was a graduate student in the late 90s, reverse genetics was all the rage, largely because we had first learned how to knock out genes really effectively in mice. And those years showed us through a lot of terrific reverse genetics. They pointed us toward pathways that many of us still study because of their importance in vascular development, pathways like the VEGF, PDGF, Tie2 angiopoietin, TGF beta pathways, et cetera. So again, terrific tools. Many of us still use these, but I should point out that the approach my lab takes sort of combines these tools in a little bit of a different way under the umbrella of epigenetics. And specifically, we study ATP-dependent chromatin remodeling complexes. And before I explain how our approach encompasses both reverse and forward genetics, I should explain what these complexes are. So these are multi-protein complexes. At the heart of them, they contain an ATPase, a catalytic subunit. And what they effectively do is they alter chromatin compaction in order to impact gene transcription. And so the way this works is that these complexes can sit down in regions of compact chromatin that are packed with a lot of nucleosome density. And using energy derived from ATP hydrolysis, because of this ATPase that they contain, they can transiently break DNA histone bonds and then start to slide or even eject these nucleosomes from regions of compact chromatin. And in that way, they can decompact chromatin. And when this happens in a gene regulatory region, like a promoter that's shown here, it can allow for transcriptional regulators, both positive and negative, that had previously been unable to access this gene regulatory region when it was all compacted. Now they can access this region, and they can thereby start to impact gene transcription, either positively or negatively. So the way that we use or exploit these complexes to study vascular development and integrity is that we specifically mutate the ATPase of these complexes in vivo using Cre-Lox technology. And we often focus on endothelial cells because these cells are so critical for vascular morphology and function. And so when we delete these ATPases in mice, we then fast forward and look for a vascular phenotype. And what we're really looking for is a phenotype that's highly tractable and reproducible. And when we find such a phenotype, that then tells us, yes, some gene or set of genes has been misregulated in these mutant endothelial cells that no longer have this chromatin remodeling capacity. And now we're getting too much or too little gene product, and that is what's leading to this vascular phenotype. So to reintroduce the ideas of forward and reverse genetics, you can see that this approach kind of encompasses both of these classic genetic techniques. Reverse genetics comes in because we have mutated, after all, a specific ATPase to generate a phenotype. And then forward genetics comes in because it's actually not this ATPase that we're so interested in, but rather the target genes of these remodeling complexes. And we can only identify those target genes by taking that phenotype and then backing up, taking candidate or unbiased approaches to figure out what genes got misregulated to lead to that vascular phenotype. So you might say, oh, well, this approach is crazy, because surely there'll be tons of genes in an endothelial cell that get transcriptionally misregulated when you mutate one of these complexes. And you're right in the sense that, yes, there are a lot of transcriptional effects from mutating these complexes. Many of them are quite small, and happily, we as a species can accommodate little changes in transcription without completely melting apart. What this approach really reveals is those genes that really do require tight transcriptional regulation. In other words, there's no redundancy built in to accommodate the little bit of transcriptional change that occurs when we take this approach, and therefore that's why you get a phenotype when those genes are misregulated. And so we're able to often prove that we've identified a correct gene or set of genes that are leading to these phenotypes because we can take pharmacological or genetic approaches to manipulate these genes and rescue the phenotype that we've generated. So this long-winded explanation of the approach we take will now be demonstrated. I want to tell you several stories that really illustrate how we've used this approach over the last 10 or 11 years that I've been running my independent lab. And you can see that we've gleaned some really interesting insights into vascular biology using this approach. So let me start by saying that we've mainly focused over the last decade in my lab on two different chromatin remodeling complexes. The mammalian switch SNF complex, which you can see contains one of two different ATPases, shown here in yellow, and the mammalian NERD complex, which contains one of three different ATPases. Now, I should point out that there are approximately 33 of these types of chromatin remodeling ATPases in the mammalian genome. So there are a lot of different complexes that we could potentially study, but these alone have provided a lot of room for discovery. So today I'm going to tell you about the work we've done specifically in mutating the BERG1 ATPase from the switch SNF complex and the CHD4 ATPase from the NERD complex. And we'll start with switch SNF or BERG. So in my lab, when we first want to start mutating a floxed allele to study its effect on the vasculature, we usually start with a Cree line that many of you use, and we start with the Tie2 Cree line. And this is because this line is really robust. It starts coming on around embryonic day 7.5 in the mouse. And it's very penetrant. It's highly expressed in endothelial cells, but I should point out it's also expressed in blood cells and lymphatic endothelial cells. So when we deleted BERG1 with this line, and I actually started this at the end of my postdoc, we discovered that the BERG mutants were quite pale and anemic, and they ended up dying at embryonic day 10.5 from this anemia. And what I discovered toward the end of my postdoc was that they were dying because BERG had been deleted by this Cree in blood cells that were developing. And it turns out that BERG is important for the proper distribution and expression of globin genes in developing hematopoietic cells. And so if you have an imbalance of these globin genes because some are not expressed to the correct levels, it makes the blood cells, the primary erythrocytes in this case, quite fragile. and they start to undergo apoptosis. And so these embryos couldn't survive past E10.5 because the majority of their erythrocytes underwent apoptosis, and this led to their lethality. Now, we were really interested in what was going on in their blood vessels because I love vascular development. And so we did find, before these embryos died, that they did have unusual vascular patterning in the yolk sac that surrounds the embryo. And we found that this mispatterning was due to a defect in the Wnt signaling pathway, specifically in the yolk sac vessels. And it turns out that BERG is important for promoting proper expression of frizzled receptors at the top of the Wnt pathway, and also of certain Wnt target genes. So it hit this pathway at multiple levels, and in general, it promotes Wnt signaling. And we could show pharmacologically that if we could rescue or restore Wnt signaling in these embryos, then we could rescue these vascular mispatterning phenotypes in these mutant yolk sacs. And then one more thing that we found from these mutants is that they had improper venous specification, both in the yolk sac and in the embryo proper. And we learned that this was because BERG is important for promoting proper expression of COOP-TF2, which is a transcription factor that sits at the top of the venous cascade. So without BERG, you don't have enough COOP-TF2 expressed, and veins start to acquire arterial characteristics because they're not carrying out their proper signaling specification pathway. So these were really interesting phenotypes. We learned a lot from the TY2 mutants, but we're interested in what other roles BERG might play beyond embryonic day 10.5 when these particular mutants died. And so we went on to use a Cree that many of you use. It's a terrific Cree. This is Ralph Adams' tamoxifen-inducible VE cadherin Cree, also called CDH5 Cree. And this is a Cree, of course, that you can turn on with tamoxifen when you want to. It's like TY2, it's highly expressed in endothelial cells, also in many blood cells and lymphatic endothelial cells. And so this Cree, we learned, if we induced it at embryonic day 9.5 and 10.5, we were able to avoid that anemia that we had seen on the TY2 Cree, but we ended up with these interesting phenotypes shown in this picture. So we got bleeding between the fingers and the toes of these mutant embryos, and a lot of bleeding in the neck region where there were large aneurysms. And these are really kind of striking and curious phenotypes. So we turned to the literature to look for anything that looked similar, and we became aware that mutants in the serum response factor pathway, the SRF pathway, looked quite similar to this. Now, SRF is usually studied for its role in muscle development, but it turns out that it's also expressed in capillary endothelial cells specifically, and without SRF, you get weakening in endothelial cell junctions in capillaries, and so they start to become fragile and they form aneurysms, which we can see here in between the toes and fingers and in the neck, and sometimes those aneurysms rupture, which leads to lethality. And so it turned out that BERG was not important for transcriptionally promoting SRF itself, but it is important for transcribing, to helping to transcribe the SRF cofactors, MRTF-A and B. And so without proper expression of these cofactors, SRF signaling is weakened, and that's why these BERG mutants ended up having a phenotype that looked quite similar to an SRF endothelial cell knockout. Curiously, if we were to induce BERG deletion with this Cree just one day later, say at embryonic day 10.5 and 11.5, we completely do not get this phenotype. The embryos look perfectly normal. So this really tells you that this regulation of MRTF expression by BERG, it happens in a very tight window of development. And after day 10 and a half, these genes no longer need BERG to help promote their expression, which is very interesting. And then sure enough, if we delete BERG with this Cree at later stages in development and even postnatally without further challenge, we end up finding no obvious phenotypes in our mutants. And so this really tells you that BERG acts early in vascular development and at least is not essential later. And this is also validated using Luisa Arispe's VE cadherin Cree. This is also a VE cadherin Cree like Ralph Adams. This one, however, is not inducible. This is a constitutive Cree that comes on slowly and is fully penetrant by embryonic day 14 and a half. And again, with this Cree, we got no phenotypes when we deleted BERG. And so this again tells you that BERG deletion after about embryonic day 10 and a half or so really doesn't have a visible functional consequence on the vasculature. Now, there's one more story though, using a Cree line that many of you have used. This is the Live 1 Cree. And this story is under preparation right now. In fact, Matt Menendez, who's moderating today, is the lead author on this. And so we're excited to get this out soon. But what we've learned using this Cree is that there is a phenotype. It's quite specific to the omentum of these embryos. And it occurs because this Cree is expressed not only on lymphatic endothelial cells, which many people use it for, but it's also expressed in macrophages and some blood vessel endothelial cells. And this story really talks about a crosstalk between all three of these cell lineages that leads to this curious phenotype of blood-filled omental lymphatics. And so stay tuned for that as we get this out, hopefully after we can get back into the lab. Okay, so some key things that we've learned taking this approach that I've described to you and from working with BERG is that BERG1 is clearly important early in vascular development, as shown through these phenotypes. And on top of that, it has some really specific temporal and spatial targets. And that was really illustrated by this SRF story where I told you that deleting it, inducing its deletion at these days gave this dramatic phenotype, which was lethal, but deleting it just one day later resulted in no phenotype. So we've thought a bit about that. And to us, it indicates, and I'll point out this is hypothetical, but it indicates that BERG1 within the switch-NIF complex may act somewhat kind of like a pioneering epigenetic factor, and that it may facilitate chromatin remodeling in order to initiate some transcriptional events that are then made more stable when you have longer term epigenetic modifications made. And of course, that could happen if the chromatin remodeling that was mediated by BERG allowed for recruitment of covalent chromatin modifiers like histone acetyltransferases that could then deposit these more stable epigenetic marks onto the chromatin. And this just points out that the chromatin remodeling that's potentiated by switch-NIF complexes, for example, is transient in nature, and it requires that constant burning through ATP. So it's fairly, energetically hungry. And for us, it doesn't make sense to have to maintain a gene like MRTF-A, which, by the way, is required throughout life to help sustain integrity of capillaries. So it doesn't make sense to us to use an energetically hungry complex like the BERG1 switch-NIF complex when you could just establish maintenance of MRTF expression through these more stable epigenetic marks. So again, this is hypothetical, but we think it makes a lot of sense given how we've seen BERG phenotypes behave. We often accuse BERG of having ADHD in that it pays attention to one transcriptional process early on, and then it sort of gets sidelined or distracted, and it starts paying attention to other things. And in that sense, it doesn't maintain transcription as much as it initiates transcription, at least within the vasculature. Okay, so with that said, let's shift. Oh, before I do shift, I should point out that it's curious to think that perhaps Berg might play a similar role in postnatal vasculature under challenge conditions. And this is something that the lab has not studied too much yet, but we could in the future. So if we were to induce Berg deletion and then start challenging animals, perhaps that quiescent vasculature, which is now assaulted, would require Berg to start to initiate changes, and perhaps we could identify some interesting vascular biology in these challenge models. So that's something that we're looking toward in the future. Okay, so let's change gears here for the second half of the talk and discuss the NERD complex. So like switch SNF, this is a multi-protein complex, and as I said, it has one of 3 different ATPases. We have primarily focused on the CHD4 ATPase. And so again, starting with Ty2-Cre, when we knocked out CHD4 early in vascular development, we got what has probably become my favorite phenotype, in that it is super penetrant, reproducible. I mean, this phenotype has looked the same way in multiple people's hands across the 11 years that I've been running this lab. And so what happens is that these mutants develop perfectly normally up until embryonic day 11.5, when they undergo this very sudden vascular rupture in the mid region of the embryo. So the big vessels running down the middle of the embryo rupture. Okay, so as we saw this phenotype, we recognized that it looked a lot like embryos with mutations in different extracellular matrix or ECM components. And so with that in mind, we started to question, well, is something wrong with the extracellular matrix in these mutants? And this led us to discover that CHD4 is really critical for regulating an ECM protease called plasmin. And specifically, CHD4 in endothelial cells helps to keep in check or repress expression of this plasminogen activator, UPA, and its receptor, UPAR. And so if you lose CHD4, you get too much of these activators, and you get too much of this plasmin activation. Now, like I said, plasmin itself is an ECM protease. It can cleave and recognize and cleave certain ECM components like fibronectin, but it can also activate a number of matrix metalloproteinases, which in their own right can cleave ECM. And so we reasoned that perhaps with too much plasmin activity, we were getting too much ECM degradation in these mutants. And I should point out that embryos at this stage of development have somewhat a still primitive vasculature, in the sense that it's a bit fragile, right? It hasn't quite recruited all the smooth muscle that it needs that will help give it strength, particularly to the arteries. And it relies heavily on the extracellular matrix to kind of help glue it together, to hold it together, particularly at this stage of development, when hematopoiesis is really ramped up, the blood volume has increased, and the heart rate has increased. And so there's a lot more blood pressure. And this fragile vasculature needs this ECM to help hold it together as it continues to mature. And so we reasoned that with ECM degradation, it might make some of these bigger vessels with a lot of pressure on them, it might make them more prone to rupture. And we were able to prove this hypothesis by genetically deleting this UPA gene that was regulated by CHD4. And by deleting it in these mutants, we were able to rescue this very dramatic vascular rupture here in the mid region of the embryo. And so that was quite satisfying to us, and it indicated, it really opened our eyes to how tightly regulated plasmin activity needs to be during embryonic development. You see, after development, once the animal is born, there are a number of proteins that are produced that help to keep plasmin activation in check. And these are things like the plasminogen, the plasmin plasmin activator inhibitors and alpha-2 antiplasmin. And so these proteins are produced specifically to help ensure that plasmin doesn't run amok, but they're not expressed until after birth. And so we believe this is why there needs to be a tight transcriptional process for ensuring that too much plasmin doesn't get made and then lead to things like vascular rupture because of too much ECM degradation. So we kind of thought that was the end of this story for the Ty2 Cre mutants, but in the process of studying them, it also led us to study a gene called RIPK3. And this came about because a colleague was helping us to look at these mutants a day prior to vascular rupture. So at embryonic day 10.5, when they looked perfectly normal by outward appearance, we started looking at them by electron microscopy and by thin microscopy. And we noticed that the endothelial cells lining these rupture-prone vessels were quite swollen, and they had swollen organelles. And in general, they just looked kind of necrotic. And in fact, that's what our colleague told us, as with his expert eyes. He said, you know what, you've got necrotic endothelial cells. And we didn't really know what to think about that until we performed some unbiased transcriptomic screening on the endothelial cells. And we saw that there was a slight upregulation of a gene called RIPK3. And we didn't know anything about that, but we could see from the literature that it was associated with a programmed death pathway, a programmed necrosis pathway specifically, called necroptosis. And so we just put the words together and said, well, here's a gene involved in some sort of programmed necrosis, which we didn't know anything about. And we have these necrotic looking endothelial cells. Maybe there's a correlation here. Maybe too much RIP3 is helping to contribute to these endothelial cells, and maybe that's making the vasculature weak. And sure enough, when we genetically deleted RIPK3 in these mutants, it rescued this phenotype, very similar to when we had deleted UPA to reduce plasmin activity. So for a long time, years in fact, we tried to draw a correlation between these two processes, the plasmin activity and the RIP upregulation. And in the end, it turned out that there really wasn't a connection that we could find. And instead, it turns out that as endothelial cells become hypoxic, which they do naturally during this stage of development, because keep in mind that the embryo doesn't really hook up to its placenta until about embryonic day 10.5. So until then, it's getting all of its oxygen through the yolk sac, and it's kind of limited the bigger it gets in terms of how much oxygen it can get. And so the embryo becomes more and more hypoxic. And eventually it turns out that this hypoxia is helping to drive RIPK3, and you really need CHD4 present in endothelial cells to prevent excessive RIPK3 transcription. Now, I should point out here that a really critical tool for figuring this out came from a hypoxia culture system that that my husband's lab purchased from Biospherics, who is sponsoring today's webinar. So big shout out to Biospherics. We couldn't have done this without the hypoxia culture chamber. And so this was really curious. Turns out hypoxia is a driver for RIP3, but too much RIP3 is detrimental to the vasculature. Now, we assumed that that meant that too much RIP3, because this has been shown in other cell types, can lead to necroptosis. And we figured that if you start to have endothelial cells dying off within the vasculature, that could lead to vascular rupture. That was perfectly logical to us. I think the big surprise of this story is that this ended up being not true. And we showed genetically that genes downstream of RIPK3 that are critical for the necroptosis pathway are not able to rescue this phenotype like reduction of RIP3 is. So it indicates that elevated RIPK3 is doing something else besides driving necroptosis in endothelial cells. We don't know exactly what that is, but we think there's some interesting hints coming from some recently published work out of Carmen Almodovar's lab in Germany. And her lab recently showed that elevated RIPK3 can help to destabilize endothelial cell junctions, in particular to mobilize VE cadherin. So we suspect that she's onto it here, that too much RIP3 is making the vascular junctions weak, the endothelial junctions weak in these embryos, and that the high pressure within these vessels is blowing these vessels apart. So I should point out that RIPK3 has become a major focus of interest in my lab. We have a number of projects continuing to study roles for RIPK3, both positive and negative within the vasculature, and whether hypoxia continues to drive RIPK3 in postnatal vessels, perhaps in situations of ischemia, and whether or not that's damaging to the vessels like it is here in the embryonic situation. So stay tuned for more of those stories over the next couple of years. So we learned again a lot using the Tie2 Cree in studying CHD4. But we did use other Crees, later acting Crees, VE cadherin Cree from Louisa, and then this live 1 Cree that I described that's expressed in the lymphatics. And this, in a nutshell, led to more discoveries about plasmin regulation and its impact in other parts of the embryo. So when we saw these mutants on both of these Crees, they looked like they had lymphatic defects. So for those of you who study the lymphatics, you'll say, oh yeah, this is classic looking. These embryos are swollen and they have blood leaking into their lymphatic network where it should not be. And I should point out, if you don't study lymphatics, that the blood vessel and the lymphatic vascular system are separate, and they really should only interface, as far as we know, at 2 spots in the body, and they're up here in the neck region on either side of the neck. And they're blown up here. And there's actually a little lymphovenous valve that occurs at these junctions. And this allows, of course, for lymph, which is the fluid that leaks out of your blood vessels and needs to get returned to them in order that your blood doesn't just desiccate out and become dried out. We need to return that fluid through these lymphovenous valves. So there's a one-way flow of lymph out of the lymphatics into the blood vessels. And at the same time, we need to prevent blood from backflowing into the lymphatics, which is not good for them. You want your blood to be circulating in your blood vessels, not in the lymphatics. So we were very intrigued to find from some work that Mark Kahn's group was producing at this time that the lymphovenous valves alone are very helpful for keeping this separation. But on top of that, there are also some small blood clots that transiently form at these lymphovenous valves. And that stood out to us because we knew that plasmin, which we knew had been upregulated in our Tie2 CHD4 mutants, plasmin is not only a potent ECM protease, but it's also a terrific degrader of blood clots because it helps to degrade fibrin. And so we reasoned that perhaps if there was too much plasmin being generated in the lymphatics of these mutants, perhaps it was degrading these little blood clots at the lymphovenous valve and allowing for blood to backflow into the lymphatics, as Mark Kahn's group had shown in other ways when they got rid of these clots. And sure enough, that was true. And we were able to show that by rescuing those embryos with these, by reducing UPA, reducing plasmin activity, and rescuing those blood-filled lymphatics. We also found in these mutants, though, that they had bloody livers. And it turned out that the liver bleeding was preceded again by high plasmin expression. This is shown here in some in situ zymography a day prior to the liver bleeding. So in this case, it turned out not to be due to high plasmin within lymphatics of the liver, but rather the fact that LIVE1 and VE cadherin are both expressed on on liver blood vessels. Specifically, LIVE1 is on the liver sinusoidal, which are kind of the capillaries of the liver blood vessels. And so in this case, again, high plasmin activity was breaking down ECM components within the liver, specifically laminin, which is a terrific target of plasmin, and which is highly enriched in the embryonic liver. And so to summarize, in this case, CHD4 again is regulating plasmin activity by transcriptionally regulating this activator receptor, UPAR. And in the case of lymphatics, too much plasmin leads to this degradation of lymphovenous valve thrombi, and it allows blood to get into the lymphatics. In the case of the liver, too much plasmin in the sinusoidal endothelial cells leads to degradation of the laminin-rich extracellular matrix around these vessels and leads to vascular rupture. And then finally, I should just point out that CHD4 does appear to have at least some postnatal roles, as we've discovered so far. We haven't fully investigated this, but we've seen consistently, multiple people in the lab have seen that when you delete CHD4 with Ralph Adams inducible VE cadcre right after birth, there are angiogenesis defects in the eye. We have ruled out that these have any relationship to plasmin. We haven't been able to rescue these phenotypes, for example, by reducing UPA. So we don't really know what the target of CHD4 is that's being misregulated to lead to these defects, but this is something interesting for future investigation. And then so together, I think these stories tell us that unlike BERG1, which I described earlier, CHD4 is important throughout development and even past birth. So it doesn't have that sort of ADHD sort of aspect to it, in that it only works at early initiating events in the vascular transcriptional program, but it maintains some of those events. Particularly, we see in the case of plasmin, which is regulated by CHD4 throughout development, although probably less so after birth once the PIs and the alpha-2 antiplasmin proteins start to get expressed and to take the pressure off the transcriptional regulation of plasmin. So just finally, I should point out that the lab does get greedy in the sense of always looking for more phenotypes. And so we have played a bit with making double mutants from some of these remodeling enzymes that we study. Early on, we knocked out BERG and CHD4 using the Ty2 Cre. Interestingly, we found that they play antagonistic roles in regulating that yolk sac vascular Wnt signaling that I told you about. So while BERG helps to promote Wnt signaling in the yolk sac, CHD4 represses it. And so together they balance, the mutants balance each other out. And so you end up not getting those defects that we saw on the BERG1 mutants. In other words, the double mutant rescues the BERG1 mutant phenotypes in the yolk sac. So that was very interesting. We've also learned that if we knock out BERG along with its alternative switch SNF ATPase Brahma, that there are some vascular defects postnatally that we don't see on either single knockout. And these are specifically lethal gut and heart hemorrhage. The bleeding is particularly bad in the gut. We don't know the mechanism behind this. This is something that the lab could potentially study in the future. I'm particularly interested perhaps in roles for RIP3 in this process, knowing that RIP is regulated by chromatin remodelers and is highly expressed in the gut. So this is an area of potential future investigation. And then finally, I should point out that we have a story that is being prepared by Melinda Wu in the lab, another postdoc, and she's knocked out Berg and CHD4 using Louisa's V cadherin-Cree. And then very interestingly, they survive up until birth, but then they very suddenly die. And as you can see, they're kind of blue, which means that they have breathing difficulties. And so it turns out that these mutants have defective lung development, which of course is driven by the lack of these chromatin remodelers, specifically in the endothelial cells. And so this is very much a story about paracrine interactions between different cell types, and we're excited as we're kind of honing in on the signaling pathway that we believe is defective and responsible for these phenotypes. So stay tuned for that as well. So just to summarize here, I just want to point out again that I've told you today about this rather unconventional approach the lab takes, exploiting mutation of these epigenetic ATP-dependent chromatin remodeling complexes, which sort of folds in some reverse and forward genetics screening to help to identify genes that are important for genetic regulation of vascular development integrity. And I would point out that some of these genes and pathways that we've been working on for the last several years are not ones that we necessarily would have thought to be super critical, not ones that we would have studied based off of their expression patterns necessarily. Some of these are ubiquitously expressed. RIP3 is widely expressed. But I think that this approach has really kind of succeeded from what I wanted to achieve, in that it has led us towards some interesting new vascular biology in a somewhat unbiased way. We've really just let the phenotypes drive us toward interesting biological processes. And it's been a lot of fun. And I see us continuing to use these approaches and then to really dig into some of these offshoot pathways more over the next decade. So with that, I'd like to conclude by acknowledging the terrific people who are in the lab right now. We've got a great group. Unfortunately, many of them are stuck at home like most of you are, but they're making the best of it, and they're thinking hard about their projects and doing a lot of writing. We've had some wonderful alumni come out of the lab too, many of whom are still in the vascular biology field. It's always fun to run into them at NAVBO conferences. And then finally, we're very grateful to the many labs around the world who make and share genetic mice resources. Many of these have been deposited into JAX, so I haven't listed those, but here are some of the mice that are currently not available through JAX that we've used. And so we're grateful to these people for sharing them, but really to all of you who have made valuable resources for our field. And of course, we're always grateful for funding from the NIH, the American Heart Association, and from local Oklahoma funding sources. So with that, I thank you very much for your attention, and I'll pass it back over to the organizers.
All right, well, thank you so much, Dr. Griffin. That was a beautiful, very clear talk. I see that we have one question that's written. Please step up, feel free to ask questions, writing them in. We'll take those first, and then we'll go and answer hands up from the participants. So I'm gonna hand it over now to Dr. Menendez so that he can direct these questions to Dr. Griffin.
Okay, great. So the first question is, are there other phenotypes on the CHD4 UPAR Tai2 Cree knockout mice? And
okay.
yeah, go ahead with that first one, yeah.
Okay, great. Yeah, so that's a good question. And you know, when I mentioned that reducing UPA could help to rescue, for example, that vascular rupture that we saw in the CHD4 Ty2-Cre mutants. While it did rescue the vascular rupture right there at that mid-gestation time point, those embryos were not able to survive all the way through development and to walk around. Instead, they tend to die from brain bleeding that emerges. We've never been able to study that phenotype yet. It would be a complicated one to study because it involves, you know, partial rescue and then generating this secondary brain bleeding phenotype. But we think that's an interesting one. We also curiously see that when we reduce RIP3 in those mutants, and so that's a phenotype that emerges either with the reduction of RIP or with the reduction of plasmin by the UPA. So it kind of indicates that neither of those processes contribute to the brain bleeding, but that there must be some other target of CHD4 signaling that gets misregulated to lead to that brain bleeding. So very, very cool phenotype, but a tough one to study just for numbers, sheer numbers of mutants
Courtney,
that needed. Yeah.
and could you speak specifically to the stiffness of the extracellular matrix on those CHD4 UPAR Ty2-Cre rescues? Is there excessive stiffness of the ECM?
Oh, that's an interesting question. So we haven't done any really sophisticated measurements like with atomic force microscopy or really any kind of engineering type measurements on the matrix. What I can tell you more histologically is that in those rescued mutants where we reduce UPA in order to try to reduce plasmin activity, we can see that the matrix components that we immunostain for, things like fibronectin that had been sowed and collagen 4 that had been reduced on the CHD4 mutants, were suddenly restored. And so this really helped to give us confidence that we were on the right pathway with the plasmin idea, because we believe that high plasmin was tearing up these matrix components, and we saw that reduced immunostaining for these components, but then that they got restored when we reduced UPA. And then just by H&E staining, as you section through those vessels that tended to rupture in the normal mutants, they were no longer ruptured. They looked very healthy and intact. So it was a very nice rescue of both the matrix breakdown as well as just the integrity of the vessel itself.
Okay. The next question is, in general, how many genes are regulated by an ATP complex? ATPase complex.
Yeah, that's a cool question. It's been shown in some different organisms, you know, going back to yeast. I should mention that these are ancient complexes, and we do find them in yeast as well as plants and of course mammals. So some of the estimates from yeast have been, you know, as much as 5% of the genome would be affected when you mutate, say, a switch SNF-like complex. Now, I should point out that many of those transcriptional effects are probably secondary. So while there are going to be switch SNF target genes that get directly impacted by the mutation, then those changes in those target genes could secondarily impact a lot of other genes, right? So the tough thing about identifying the target genes is that you really have to do it by chip. There are no consensus binding motifs that we can use to predict where these complexes are going to sit down. And so instead, people run a lot of chip-seq type experiments to try to understand within a certain cell type, what are the genomic places that these complexes sit down. And of course, I would argue within an endothelial cell, you know, endothelial cells are so unique in terms of their location and how they behave at different times in development. Just because you've identified, say, the chip-seq pattern in one type of endothelial cell, say a HUVEC, doesn't mean that necessarily has any bearing on, say, the liver endothelial patterning of where these complexes would sit down at E14.5. And so for this reason, our lab has leaned not so much on genomic type approaches to study these targets, but this has been our justification for leaning very heavily on the phenotypes to drive our biology and to really point us toward critical times and places that these complexes are doing things that really functionally matter in terms of the vasculature.
Okay, the next question is CHD4 expressed in all vascular beds during development?
Oh, okay, yeah, this is a good question. We think so, as far as we can tell. But you know, that's only as good as your immunostaining is going to be, of course. I will say in terms of its expression that it and Berg are both downregulated during the course of development. CHD4 in particular, we've looked at, for example, within the liver vasculature, and it gets dramatically downregulated throughout the course of development. And so this tells you that these chromatin remodelers, and this is borne out by a lot of our phenotypes, are heavily involved in developmental processes. And then after development, of course, they're going to still play roles in transcriptional regulation, but maybe not quite as critical roles. And that's certainly shown through the lack of phenotype in our BERG1 mutants that we generated after birth.
Okay, so the next question is, has there been a postnatal role assigned to the chromatin remodeling complexes in postnatal angiogenesis, specifically tumor angiogenesis?
Okay, so this is a perfectly fair question. We have only thoroughly studied it, and I should say we've only really looked at tumor angiogenesis under the BERG1 mutants. And so we did that using Lewis lung cell carcinoma models, which many people in the field use, LLC models. And we did some Matrigel plugs as sort of a proxy way of trying to generate a tumor-like environment. And in both of those cases, we saw no defects in terms of the angiogenic process of either growing tumors or investing vasculature into the Matrigel plugs. And so for this reason, we have said, at least in those models, that there's nothing obvious, there are no obvious impact of Berg on tumor angiogenesis. Now, those are limited, right? Those are just two models, and so perhaps there are more specific types or models of tumor angiogenesis that we haven't yet explored that may find some interesting effects in Berg mutants. But so far, we haven't found any, and we haven't looked at CHD4 yet in this context. But these remodelers, I should just point out as a side note, are very important in cancer biology in non-vascular cell types. And so they definitely modulate tumor cell proliferation in different types of tumors, and they're widely studied in the cancer field.
So how does ECM failure in the liver lead to leakage?
Okay, that's a cool question. And we had to do some deep diving into kind of anatomical descriptions of how the liver vasculature develops. So you would think that these sinusoids that I described, these little capillary blood vessels, don't have a lot of pressure, right? And yet we always saw bleeding initiate in those CHD4 mutants in the left lobe of the liver. And so as we dug into the old kind of anatomical literature, we found that the left lobe is where blood shunts in from the placenta through the umbilical vein. And so that particular lobe of the developing liver at this point in mouse development is under higher than normal vascular pressure. And so that's why we think we particularly see bleeding in that site. So it kind of gets back to that idea that the ECM is helping to hold together a somewhat fragile vasculature, particularly under situations of higher blood pressure. And we think that that left lobe is such a situation where at this time in development, there's a higher than normal blood volume and pressure that's flowing into it. And that's what makes it prone to rupture.
And can the lymphatics in the liver pick up this fluid?
Okay, interestingly, we didn't find great evidence of lymphatic development in the liver at this stage. And so we think liver lymphatics develop later. So I think for this stage of development, no, that there couldn't be a great pickup by lymphatics, at least from within the liver itself. They're of course developing outside and around the liver, but from within the liver, we don't find great evidence of their development this early, at least by the markers that we've used to study them.
Okay, I think that is all of the questions, unless I'm missing any.
We have Barbara Summers, who has one more question. I think we need our...
Okay.
Okay, great. Barbara, go ahead. I don't know if you still have a question, Barbara. I see your hand up and you're unmuted, so you're welcome to go ahead and ask. Okay, well, listen, I think that maybe we're out of time, and so maybe we'll close it out there. So I really hope everybody found this beautiful research presentation beneficial. Just remember, these are brought to you by the NAVBO Education Committee. And I just want to say that our next webinar is on Thursday, May 14th, same time, same place, this time with Martin Schwartz of Yale School of Medicine, and he's going to be talking about matrix remodeling and integrin signaling and endothelial function and dysfunction. So look for all that information in the NAVBO newsletter. And so if you would, please, we would really ask you to go and fill out the GoToWebinar evaluation when you receive a subsequent email, and let us know what you think and what we can do better and the things you'd be interested in for future NAVBO seminars. So thank you all, and thank you, Matthew, and especially thank you, Dr. Griffin, for the beautiful presentation. So have a great day, and everybody stay safe, stay home, take care. Bye, everybody.